Welcome, everyone. Thank you very much for the kind introduction. So I'm very happy to be here. It's the first time for me at the summit. As you can see, so we have really wonderful experts here for our genetics panel, and the five of us, we will try to walk you through where are we at the moment regarding genetics, what is the uptake, what are the main barriers regarding adoption and incentivization. So I will start with the first introduction of the Global Methane Genetics Initiatives that we are running in Wageningen. So the Global Methane Genetics Initiative is funded by the Bezos Earth Fund and the Global Methane Hub and coordinated by Roel Werkamp and myself, Wageningen Livestock Research in the Netherlands. But before we go into more detail, I would like to set the scene a little bit. Where are we in terms of low methane genetics? We do have recording techniques available that allow us to record a large number of individuals for methane emissions. I'm not going into detail in all those different techniques, so there are pros and cons, and I'm sure you will have different opinions on that, but they exist and allow us to record methane on the animals. Second, genetic variation for methane emission exists. So several studies have proven that methane emission is moderately heritable. So that really is the base so that we can use genetic selection really to gain genetic progress in the trait. And this has led that breeding values for methane emission are no longer a future concept, they are reality already. So I've put some country flags here. So some countries have already implemented breeding values for methane genetics. So the question is really no longer whether genetics works. So it's really about how can we scale it up, how can we implement it, and how can we maximize the impact. And this is, I think, where the Global Methane Genetics Initiative come into play. The main aim is to accelerate genetic progress for lower methane emission in ruminants. I've brought the key figures here so you can see it. It's really a large international effort, more than 50 partners, and the overall aim to phenotype more than 100,000 individuals. The core activities really span across the whole pipeline. So from phenotyping, data sharing, genetic evaluation, breeding program design, and ultimately adoption and impact. And then along the way, we develop protocols and standards for all those steps that I've just mentioned. One key element is that for breeding that we need large reference populations. And I've brought this map here to show you the consortium of the Global Methane Genetics Initiative. On the left-hand side, you can also see the projects that are running within the initiative. So it's 7 projects, and the numbers give you an indication of how many individual animals we are going to phenotype within those projects all over the world. You may see that there was a missing spot on Asia. So just recently, we welcomed a project within the initiative on small ruminant smallholders in India and Mongolia and Tunisia. And then, in addition, we are working on proposals for buffaloes and Asian cattle. So bringing all this data together really needs an effective way of use the data and share the data. So very briefly, I would like to mention the Global Methane Genetics Database, and we are very proud. So it will be going online soon. It's been tested. So all these data that will be collected within the initiative will be made available for sharing to really accelerate genetic progress in methane emissions. So it's really that none of us has enough, basically budget or so to do it all our own. So we make this data available based on a fair share principle, build trust between the partners, but also, I mean, it's open for any third parties to contribute here to create larger reference populations, better breeding values, and ultimately lower emissions. When bringing data together from a large number of partners internationally, so we need to really think about how to do this, how to bring this data together. So we are closely working here with the ICA Feed and Gas Working Group, and I'm happy to chair this group, and which is really a nice collaboration within the GMG initiative and the group. So we're setting up the protocols and standards together, how to record, how to analyze the data, compare pipelines and so on. And the nice project that we have just recently completed within GMG and ICA is that we have set up a methane device test center at the dairy campus in the Netherlands, so where manufacturers can actually get their methane device tested and validated. So having all these data collected, so now it's really coming to the implementation side. What are the real gaps or the issues that we are facing at the moment? So within GMG, so we have also set up a project covering issues or gaps that we see regarding adoption and incentivization. One key point is validation of methane breeding values. So we've developed an open source protocol to really prove and get confidence so that the breeding values really deliver methane reductions. Second, trait definition and breeding objective. So dose is really a topic that heavy discussions on which trait should be used, and it needs to be clear what are the consequences of using those traits in our breeding objectives. And third, very important, policy levers and market mechanisms. How do we create incentivization of genetic selection as a mitigation tool? At the moment, no economic weight exists for farmers. So farmers really need to have an incentive why they should be taking methane breeding values into account in their selection decisions. And with that, key messages that I would like you to take home today. So no one should leave the room without believing genetics works. Global infrastructure is underway. Validation is underway as well. And we're working on adoption and incentives for the next frontier. Still, we need to keep in mind trade-offs and the economics as key priorities. And with that, I would like to thank you for your attention. And moving on to the... And we are continuing now with the next presenter, who is Guillermo Martinez Boggio. I hope I did it correctly, pronounced the name correctly. The floor is yours.
Good morning, everyone. Thank you for the invitation, Joe, Frank, and all the organization of the summit. It's a great pleasure to me to be here today. So, where is the clicker? There you go. Thank you. So today I will try to answer this question that is talking about what happened in the last 50 years about methane emissions. So this is probably the first picture that we show to students in animal biology, in animal breeding and genetics. And is the genetics works, as Birgit mentioned just before. A cow today produces roughly twice as much milk than in 1960s. And that was the result of systematic selection combining genetics and better management. Genetics gain are permanent and cumulative. So when we talk about reducing the environmental footprint of the dairy, genetics is a very powerful tool. What drives this genetic improvement was the flagship of the dairy industry, that is the Lifetime Net Merit Index, an economic selection index that is used to rank dairy bulls and cows. This definition of the index evolved across the time. So we were in 1971, when this index was developed for first time, we were interested on cows that produce more milk. And then in the 1994, we start to want a more balanced index. We want cows that produce a lot of milk, but they are healthy, they are fertile. And then since 2020, that was the first inclusion of the residual feed intake. We are selecting for more feed-efficient cows. So the question here is what all this selection produce indirectly on methane production. The result that I will show today will not be possible, but because a multi-institutional, multidisciplinary research group led by Professor Peña-Ricano at UW Madison in 2023 start to collect data from research and commercial farms. Today, June 2026, 3 years later, we have more than 3,000 cows and over 4,000 lactations with methane emissions, intakes, body weights, milk yield, and milk composition. And this data will be the beginning of the reference population that the Council on Dairy Cattle Breeding will use to develop the first genetic evaluation for methane emissions in the US. The data is exceptional, but it's not enough. We need to keep collecting data from different environments and different production systems. From this reference population, less than 5% of the data is coming from California dairies. And that is what I'm here at UC Davis. One of my main goals is to bring data from California dairies to the genetic evaluation. So as Birgit mentioned, we have methane emissions, methane production, we have a heritability of 30%. That means that methane production is controlled by genetics. We can do progress. And as you can see in this chart, it's at the level of the milk production and the fat yield. So if you think all the progress that we made on milk production, imagine what we can do on methane production. Also, what drives methane genetically? Those are some of the genetic correlations that we have. Methane production is highly correlated with fat. What makes sense because the fat synthesis is tightly linked to the rumen fermentation. In the other side, methane emissions are highly correlated with feed efficiency. That means that if we have more feed efficient cows, we have cows that produce less methane. So those correlations can give you an idea of what it happened in the last 50 years indirectly to methane emissions. So if you see in this chart, you have a first phase that started in 1971, where all the emphasis was placed on production. We want cows that produce more milk. And that means that we increase methane production. After 1994, when we start to have a more balanced index, we start to include fertility, health traits, longevity traits. We start to have a lower pressure on methane emissions. But the inflection point is after 2020 with the first inclusion of residual feed intake in the index, what makes methane goes down. But still we are in positive numbers. So we have positive correlate response. So this indirectly effect is not enough. We need to go for more. So as a takeaways, genetics is promising because methane emissions are heritable and we have large variability. Second, the improvement that we make using or the evolving definition of the index is good, but it's not enough. And third, what we need is to include methane emissions on the selection index to start to select directly for methane emission, and in this way we will make much more progress. But for that, we need data and data from different kind of environments. Thank you very much.
Thank you very much, Guillermo. Just a reminder for the audience, yeah? So you can already start putting your question into the Slido, so you can address them then afterwards in the discussion. So I'm happy to introduce our next speaker, who is Matthew Cleveland. Matt is a Senior Director of Global Bovine Sustainability at ABS Global.
All right, thanks, Birgit. So, let me start. Oops. All right. So I work for a livestock genetics company, just in case you don't know who ABS is. And as you know, we're thinking about these new traits and new technologies. One of the things that I spend a lot of time thinking about is how do we scale up these technologies, right? As a commercial breeding program, how do we implement? How do we scale up? And ultimately, how do we drive adoption, right? And adoption in commercial production systems with the massive amount of variation around the world can be challenging. And so one of the things that we really think about is, the fact that really scale is our key to really delivering impact. And so I'm going to spend a few minutes today, the rest of my 5 minutes here, talking about, what does that mean? What are some of those key factors that are going to affect how we scale up new tools and new technologies? I think one of the key things that we've already heard about, and I think we know, but production systems that aren't profitable aren't sustainable. And so anything that we implement has to add value to the system and not just add costs. And one of the ways that we can drive some additional value into the system is through incentives, right? Oftentimes farmers want to do the right thing, but there's a lot of conflicting priorities in their operations. And so they oftentimes need clear incentives. They need clear signals about what to produce and how to produce. Incentives, they're going to drive behavior, but at the same time, implementation of the genetics has to be tailored and optimized to the system in order to have an impact. Again, we have lots of variation in the types of systems, and the tools that we use really need to be optimized for those different systems. And then ultimately, those innovations need to work within the current systems, and they need to integrate well, right? We need to have the ability to seamlessly implement new tools and new technologies. And so really for new technologies, innovation to be truly impactful, we have to create value at farm level. And we have to be able to align with value chain needs. And really what this allows us to do is scale vertically. But at the same time, it's really important that we can scale horizontally as well. So again, if I have a new genetic tool or new genetic technology, I need to be able to scale that widely across different types of production systems around the world. And I think a lot of the genetic tools that we have and that we're developing are well positioned to do this. But effective implementation really requires a set of key enablers. And we heard Rory last night talk about the use of artificial insemination. In an agricultural meeting, I used to be able to say AI and people would know what I'm talking about, but now I actually have to go back in time and actually say artificial insemination, right? But effective implementation requires some of these key enablers. I don't have time to talk about these today. There's a lot of different things we could talk about. But I think what we have to realize is that some of these enablers, again, something as simple as artificial insemination, they're underutilized, right? They're not optimized, even in developed production systems. And so our challenge is, how do we enable farmers to use the tools we already have, as well as the tools we're developing in an effective way? And so I just have a couple of examples in my remaining time to talk about what this can look like in practice. And so in a beef on dairy production system, for example, we evaluated the environmental impact of a targeted breeding program. Again, one of our key enablers. We call that program our newer genetics program. But it's a targeted breeding program for beef on dairy systems in the US and the UK. We evaluate the environmental impact through life cycle assessment using 3 different genetic backgrounds. And so essentially what we're doing is we're making genetic improvement at a nucleus level for traits that drive profitability in a beef on dairy production system. And then we're disseminating that improvement to dairies, in this case in the US and the UK, who themselves are implementing key enablers, right? They're using sex semen to create their replacement dairy females, and they're using some proportion of beef semen on top of that in order to optimize their own productivity. And we found in this work that the overall environmental impact was reduced across 16 different impact categories. Now, I'm only showing the climate impact category as it's a little more relevant to our discussion on methane here, but we were able to see a decrease, which when I accumulate that, genetics are permanent and cumulative, when I accumulate that over time, starts to create a material impact or material effect on that reduction in environmental impact. And so in this case, we've demonstrated that through this type of production system, we can improve system profitability, we can improve productivity, while at the same time reducing environmental impact intensity. And again, because of that cumulative and permanent nature, we'll see those effects over time. Another example is from dairy production in the UK. This is very initial work, but we've evaluated 170 different farms after implementing programs to optimize what we would call kind of their genetic management system, right? Optimize the amount of sex semen they're using to create replacement females, the amount of beef semen they're using, use genomic tools in order to select the best replacement heifers and evaluate their herd age demographics. And what we saw was that from 1 year to the next, after we implemented these changes, we saw a material change in the absolute or in the average index values. And this is an index PLI that's used commonly in the UK. But what we saw is that index on average went up more than would be expected from the annual rate of genetic gain. And you can see there's a lot of variation there. Some farms did mostly, they didn't do worse than they did the year before, but some farms did materially better. And what we know is that this index is directly related to reduction in environmental impact. And so this is a case where we didn't create new traits, we didn't create new genetics or new tools. We're just optimizing how those existing genetics are implemented. So even in developed production systems like in the UK, there are still opportunities to drive productivity. And this is very preliminary. We're working today to really understand what is that true environmental impact. So as I wrap up here, I see I'm out of time. I think ultimately, what I really want to get across is that, we're looking at how do we scale up these new technologies. You're going to hear a lot today about new things that are happening in the genetic space, but getting them into commercial production systems can be really challenging. I think there's a lot of opportunity here for innovation, whether it's new traits or new technology. We continue to invest and work in this space, but we also are here. We're here in this meeting talking about the direct improvement in GHG emissions, but there's still massive opportunities for continued productivity gains. And of course, this is going to impact emissions, right? We know that we have not optimized production systems around the world. And so implementation of these tools, still continues to lag, but by optimizing the systems, we're going to drive productivity, which is going to reduce environmental impact, but also position ourselves better to implement and leverage the tools that are being created. And with that, I will wrap up.
Thank you, Matt. So I would like to welcome our next speaker, who is Mike Lohuis, who is the Vice President of Research and Innovation at CEMEX, a global dairy and beef genetics cooperative. Mike, the floor is yours.
Thank you, Birgit, and thank you to the organizers for the opportunity to tell our story. We started this story in 2023 when we first introduced low methane genetics in Canada. It started with a lucky happenstance. The researchers at the University of Guelph discovered a connection between actual methane measured from green feed feeders with milk spectral data coming from milk samples. So this was very lucky for us because we learned that there was a phenotypic correlation of 70% between the milk sample data and the actual methane from the cow. At the genetic level, it was a 92% correlation. So when you're thinking as a geneticist, how can I improve a trait that's expensive and hard to measure and takes a lot of time, it's good to have a proxy trait. So we're using a combination of these two sources of data, direct and indirect information about methane. And it was laid out in a paper in Dairy Science on the methodology. So we went back a couple of years later to look at a separate set of animals, also in a research environment where we're measuring green feed. So these animals were separate from the original reference population where we generated the breeding values. And we were able to see about a 10% difference between the high animals and the low animals. High being the top 16%, and the low being the bottom 16%. So about a +1 or -1 standard deviation from the mean. And one of the things that we're we've done to make it easier for farmers to understand is we flip the sign so that a high animal is actually more methane efficient, whereas a low animal is less methane efficient. So more methane per unit of milk, for example. So this was a validation done in Canada, but we wanted to also see, does this replicate on the international scale? Because we're a genetics company, like Matthew and ABS. CMEX is exporting semen and embryos around the world to 80 different countries. So we wanted to make sure this translated in other countries as well. So we were able to exchange data with France and Italy and the UK. And these were animals that were both genotyped and had actual methane measures from green feed. And we were able to see about, similar to what I just showed you, this is about a 9% difference between the high animals and the low animals. So it was statistically significant at about the 1% level. So it was nice to get that verification that it works not just within the home country, but also internationally as well. So differences between high and low animals in the current generation is valuable and it's interesting, but it's unlikely farmers are going to cull their cows because they're producing more methane, unless there's a strong incentive to do so. What's more important is going forward, and as Guillermo showed in his first slide, that genetics does work. So if you practice genetic selection in a systematic way, generation after generation, you can expect to see a significant change over time. The orange line shows what the actual equivalence would be for CO2 equivalents for a typical dairy cow in Canada, and what would that decrease look like over 25 years. And the gray line is showing what would be the cumulative reduction. So the assumption in this, this is a gene flow model where we looked at selection of bulls that was 1 standard deviation or more above the mean. And so we also looked at female selection that is above average. Right now, most dairy farmers are using sex semen, which allows them to generate the replacements from about the top third to the top 50% of their herd. So these are fairly reasonable, not very taxing restrictions. So what does that add up to over time? It's about a 36% reduction, 1,350 kilograms of CO2 equivalents per year by the time you get to the end of this period. The question we often get asked is, well, are there any genetic tag-alongs, anything that happens that you didn't expect? We don't have genetic correlations per se, but we do have correlations between the breeding values that we do collect. And you can see that there's not many correlations different from the mean, at least significantly. The only two that are significant really are metabolic disease resistance at the bottom and health and fertility. And so these are favorable correlations. We didn't see much in the way of negative correlations, nothing at least that's statistically significant. So that was reassuring to us, and this is something that you can monitor over time as well. You can select for feed efficiency as well, and you can select for body size. Both of those traits will also reduce methane efficiency. So to summarize, methane can be reduced about 1 ton of CO2 equivalents per year per cow. And over the Canadian population, we have about 900,000 milking cows and 1.1 million head of dairy cattle. We could reduce emissions by about 1 million tons of CO2 equivalents per year for Canada. And predicting methane using MIR or mid-infrared spectral data has a high reliability for 70% reliability for cows with only a genomic sample. Genetic change, unfortunately, starts slowly and takes time to cumulatively add up to those reductions, but it is permanent and you would have to select against it to reduce that. One of the things that we do like is we can prove that it is additional to the current practice or business as usual, and it is causal. So there is a causal change in methane reduction by selecting, and that's something that verifiers want to see. The unfortunate part is that dairy farmers are rational human beings and they do what they get paid for. And right now there's no economic incentive for them to do this. So we need to provide an economic reason for them to select for this because, as was mentioned, genetics does work. Thank you.
Thank you very much, Mike. I'm happy to introduce our last speaker in this round, who is Emily Melchior. Emily is Manager of Global Outcomes Research at Zoetis Animal Health. Please welcome Emily.
Good morning. It's a wonderful pleasure to be here to speak with all of you today. I'm very excited to share about some of the sustainability enhancements that Zoetis has invested in. It's something near and dear to a lot of people's hearts, but ultimately we are here for the producer. They are producing feed for the world. They're producing a product that is hopefully going to go and sustain humans throughout the world. So we're ultimately here for them. So I wanted to give a little bit of context about what dairy wellness profit is. This is our selection index that is informed by over 26 different traits, where we place a significant amount of value, of course, on production, but it really encompasses the whole animal trait. It's not just looking at her milk value. It's looking at how does this encompass some of her health traits? How does she respond to antibiotics? It's looking at lameness factors, metritis, mastitis. And one of the things that we felt was an opportunity for another enhancement was to look at how does that influence her sustainability within the herd. And not just within the herd, but her sustainability, of course, in producing milk and remaining as a productive member of the herd. So the update for 2026, of course, we still have our emphasis on some of our top 5 traits of importance, including fat, protein, mastitis, body size, and lameness. But we added an additional trait that really helps inform this index to make it a more sustainable alternative for dairy producers. And that is this trait at the bottom of the screen that's called rumen, which stands for rate unit of methane in nature. And this trait was informed by our recently published data, supported through the Journal of Dairy Science in conjunction with Dairy Management, Inc. and some of our other colleagues within Zoetis. And so ultimately, this is our enteric methane prediction that has been validated and published. And that's truly what's informing these traits moving forward. So if you're not really familiar with DWP, you might not be familiar with how it works. So in the case of millions of records that we've obtained over years, the best 25%, if we break these animals into quartiles, the best 25% of those animals via DWP ranking are going to outperform the lowest ranking DWP animals substantially, up to $800 more in profit per year or per their lifetime per animal. And that's a considerable amount of money that producer is able to utilize while they're improving genetic progress. But it's not just about the milk production. We also wanted to see how these traits are influencing some of the additional factors that go into an animal's health and well-being. So looking at mastitis, lameness, and metritis, we had the same animals that are already producing more from a production standpoint and being more profitable, but how does that also contribute to their health and well-being? And you see that on the far right-hand side of your screen, our top 25 producing animals are having significantly less mastitis, lameness, and metritis events throughout their lifetime within the herd. So not only is this an opportunity for sustainability within some of the enteric methane predictors, but also you're having to apply fewer interventions to keep these animals healthy, productive, and within the herd. So for the sake of time, while we have 6 updates to share with this group, I'm going to focus primarily on 2 of them, but I'm happy to chat with folks after the fact if that's of interest to them. And for the sake of this, I'll focus on milk methane intensity and DWP heat. So our milk methane intensity prediction was developed in commercial herds across 5 different states with over 13,000 animals that were enrolled. And these animals had the opportunity to use a green feed machine, I think of which most of us are familiar with. And we tracked their milk production throughout their lactational period. So this is all traits that were developed on commercial dairies. These aren't a Zoetis research herd. And the majority of this data was collected during the peak lactation interval, but it does represent the full lactation period. And what we found is that this milk methane intensity prediction really demonstrated that these animals do have an ability to have reduced methane output per kilogram of milk produced. And so when we inform to producers how this looks when they receive a genetic audit from us, is that they will be able to look at these numbers and say, this animal is being a little bit more efficient. Whether they're being paid on components or being paid on a fluid milk percentage, they can use this to help inform some of their decisions. But ultimately, we also have the responsibility at a global level. And heat stress is something that many of us are familiar with, particularly this week here in California, it seems. But because we are interested in the overall health and well-being of these animals, we also have to consider that they're facing heat and humidity stressors that they might not be necessarily facing in other parts of the world. And so in order to compensate for that, we developed DWP heat, which is an additional enhancement through our Dairy Wellness Profit Index that really looks at the opportunity of the heat resilience of these animals that we've identified through fertility heat resilience and milk heat resilience. And so ultimately, when these animals are experiencing heat stress identified through a temperature and humidity index of at least 20% of the days of the year, we wanted to give an opportunity for producers to really give credit where credit is due. They're still able to be productive, they're still able to be within the herd, and that's informed by their ability to have higher first service conception rates, as well as milk production through these heat stressor events. But ultimately, we also want to make sure that we're not sacrificing production for any of this. So even the producers that might lean towards DWP heat as an option for their herd, they're still going to be making improvements in terms of quality production, improvements toward antibiotic stewardship and welfare, fertility, and precise nutrition. So ultimately, these are just options that we can provide to producers to help them really influence their herd. Genetics works, as the other colleagues have said today on screen, it really does work. And this isn't a multiple time point intervention that has to occur. This is a single intervention that producers can use that they don't have to constantly administer. So it is a sustainable alternative to making animals healthier and more productive, and that's what sustainability should be. Thank you.
Thank you very much, Emily. So I now invite all the speakers to come on stage for the discussion. Matt, Mike. Working? Yes, great. So, well, thanks all for the very nice overview presentations. What you're all doing in the field of greenhouse gas emissions, methane emission. We already have very nice questions, I would say, from the audience in the Slido. So I think we just get started with questions from the audience, which are really very relevant. And there are 2 questions actually dealing regarding trade-offs. Maybe can every one of you comment a little bit on what are the trade-offs, or how do we look at animal health? How do we include it in our breeding programs? What are genetic correlations? What do we need to do, basically? Would you like to start, Guillermo?
So I think that as I show in the presentation, so the inclusion of methane. So first we need to define which trait, that is another question that should be around, is which trait do we include in the index. if that is methane production or should be any of other available traits like residual methane production. But also what we need to look at is the genetic correlation, so what we will move down or up, because in an index that is the play that we need to have is we will put more weight on some traits, we'll reduce the weight on some other traits. So what I show, for example, a next step could be to put less weight on fat because it's highly correlated with methane production. But also there should be a discussion if that will be the next step or what we want to do for the future. But for sure, the two traits that are moving methane up or down are fat yield, that is moving methane up, and then from another side is feed efficiency, that is moving methane down. So more efficient cows produce less methane.
Yeah, we ran into this trade-off problem early when we first produced methane evaluations, methane production. And what we immediately saw was this negative correlation with fat. And in Canada and in many countries, farmers are paid based on fat content of their milk. So that was a bad thing. Farmers would immediately say, why would I select for something that's going to reduce my milk check? So we went back to the drawing board and reconfigured the trait to be independent of milk, fat, and protein. So that was what we called methane efficiency, different from methane production. So methane efficiency, we could tell our customers, does not affect production traits. From our genetic studies, we've seen that there's no significant correlations really that could change over time, but for now, we don't really see any drawbacks on other traits. The biggest consideration might be if you were to put extreme selection on methane, what would you be giving up in terms of all of the other economic traits that you want to select for? We did a little study that showed if you were to select bulls that were 1 standard deviation above the mean, how much would you give up in the overall index? And to our surprise, there was no reduction in the two formula for the two. We used Pro Dollar and we used LPI, which were the national indexes in Canada. And they didn't have to give up anything. It was just a smaller list of bulls, that's all. So you had fewer bulls to choose from. So that's maybe the biggest drawback at this point, is you have fewer bulls to select from.
Okay, I think it's on now. I think, especially when I consider some of the selection with the DWP heat that we've been integrating as an option for producers, there are inherently some trade-offs. A more heat resilient animal is probably not going to be your highest producing animal, but we don't really want to sacrifice that. If she's more heat resilient or she's able to withstand some of those temperature and humidity constraints, we don't necessarily think that's a bad thing, that maybe her production hasn't spiked or hasn't peaked like an animal that's not experiencing heat stress. So there's, of course, going to be trade-offs no matter what trait you're selecting for. But if it's more profitable for that producer to maintain that animal who, for example, can maybe handle some of that heat stress a little bit better, even if she's not maybe as productive as some of her counterparts in cooler temperatures or cooler parts of the world, we just want to give that opportunity. And that's a pretty, particularly with their heat stress traits or heat resilience traits, the more heat stress that animal is facing, the more heritable those traits are. So I think that's interesting to look at. So, but depending on what that animal or what that producer is faced with, they're going to have to make these decisions. And we just need to be able to provide tools and evidence to support them in that decision.
And I don't think I have a lot to add. I mean, maybe I'll step out of the dairy space a little bit and think more beef, where I think we understand much less. And I think there's a lot of work to be done, particularly on the health health and disease side of things, right? I think just from a traditional breeding program standpoint, there's a lot we don't understand in beef improvement around health, and I think then you add the methane piece on top of it, and that just adds to the things that we don't understand. So I think there's still a lot of work to be done in both spaces, but I think beef has a much longer way to go in terms of just being able to refine how we define animal health, and what that actually means.
Thank you, Matt. Well, good to have consensus that, well, it is important to look at the trade-offs or look at the correlations to the other trains, and and we're all doing this. So. Moving on, changing topic a little bit. A question that is highly ranked in the Slido, and what is a topic that has been mentioned also in almost each of the presentations, I would say, are the incentives, adoption and incentives. So what are the chances that low methane genetics could qualify for credits? Is there a methodology already in place? This at the scale that ABS, specific question for you, Matt, manages could substantial even at the 10 to 15% drop. Would you like to start?
Yeah, right. I mean, I think ultimately there is certainly an opportunity in genetic improvement, right? We talk about genetic improvement or genetic innovation, being a key intervention for reducing environmental impact. And so I think we can look at genetics as an intervention or as a practice, just like we look at a number of different practices that are being incentivized to implement. I think, even though the numbers appear to be small, when we look at sort of the size of reductions we're talking about compared to when we look at things like feed additives and those sorts of things, what I think we all talked about is the fact that these changes are cumulative and permanent, and so the reductions we see, they're going to accumulate over time. And so I do think there are certainly opportunities across the supply chain to create incentives to reduce carbon, reduce environmental impact. Now, what does the framework look like, and how do we do that? I'm not sure that that's clear. At least we certainly don't think it's all that clear. And whether this is going to survive solely as an inset opportunity, whether there are going to be opportunities on the offset market for avoided carbon, I think that's still developing.
Mike, would you like to comment as well?
I think we're getting close. One thing that's really nice about genetics is that it is permanent and cumulative. So one of the things that verifiers want to see is that you do have a permanent solution, that you won't backslide. You also have to prove additionality. One of the things we did was we looked at methane efficiency and what was the genetic trend over time. And when you correct for milk, fat, and protein, the genetic trend was 0. So unless we do something, it will continue to be 0. So that's a way to prove that this will be additional to current practice. And we also have to prove that if when you do make these mitigations, that it will be causal to show that you've made an actual change. So I think we're getting close to answering those questions. Of course, you need to have the peer-reviewed literature, and that helps give everybody in the supply chain, it takes the risk out for them that they know that there's peer-reviewed literature that shows. I guess the other part, given that genetics tends to be done on a national basis, that if you can establish a genetic prediction, it is based at the national level. So it's not based on one farm, it's based across all farms.
Maybe just one small thing to add based on your comments around kind of additionality and permanence. I think one of the challenges, and Rory mentioned this last night, one of the challenges we face is that the frameworks that are being developed at the international level don't necessarily take food production or production agriculture into account, at least in a way that we might like to see it done. And so there's some creativity, I think, that we're needing to develop to sort of get there, assuming that these sort of international frameworks are going to come along at some point.
I'll take one comment that you just made before, Matt. So there are other interventions that already made it basically into the calculators. So very short answers. So what is really the key? What do we animal breeders miss? What do we need to do?
We need to just explain genetics better, I think.
Yeah, maybe it's the communication. I think that's actually a very important part. Yeah.
Yeah, so go ahead. Well, I was just going to say, I mean, I talked to a lot of supply chains, right? And I was talking to a very large global supply chain a couple of months ago in the animal protein space, I'll say. And one of the things that they told me was, you know, we now believe that genetics is the most important thing we need to be focused on in order to meet our climate commitments. We just don't know what to do, and we don't know how to do it.
Yeah, I'll just echo with the communication piece, something that I think is maybe undervalued, and as scientists are being trained and such, is how important it is to truly communicate the good work you are doing and how capable it is at making change. And so having that be a more robust part of the discussion, I think in the future would maybe make this a little bit easier. Not easy, but it would make it maybe a touch more easy.
Changing topic again a little bit. So there's one question from the audience that is highly ranked again. I'm sure you probably all are aware of the study in New Zealand where they're comparing daughters from bulls that were selected based on methane emissions. And they recently just released results of this study from this large experiment where they bred cows to low methane bulls. So where the daughters produced are in their first lactation and there are no differences in methane production. Are those results inconsistent with the global evidence? Who would like to start? Guillermo, would you like to comment?
Yes. So I was not aware of this work, but something that I want to remark here is, and probably it's important to mention that when we are talking about, or at least when I'm talking about genetic solutions to reduce methane emissions, I'm strictly speaking to develop a genetic evaluation. So farmers in the future, they will be able to select their bulls based on how much methane they produce. And that will be only possible if we have a well-defined genetic evaluation. And for that, we need time and we need phenotypes, because we cannot collect thousands and thousands of phenotypes from today for tomorrow. So we need years of phenotyping in research and commercial farms in different environments. So the environment the diet in the Midwest is very different of what we have here in California. So if we develop a genetic evaluation only with data from the Midwest, so probably this genetic evaluation will not work for the dairy farmers in California. And that is why I want to be very cautious about talking work that other people is doing in other countries, because I don't know how they were developing their genetic evaluation. And that is probably the question that you have at the end of each summit. And I think that will be my second summit. And then I attend the first one virtually. Always we are saying genetics is a promising tool. And we leave the summit and we say, genetic is promising. We will have a very powerful tool. Well, we are working on that. So we are collecting phenotypes. We are working on developing a genetic evaluation that brings reliable results for farmers. And when they make genetic selection, they have the result that we expect and they expect.
I'm not sure if it's the same study, but I remember there was a study recently release that showed measuring methane on bulls, right? I think this is the same study, and then looking at the results, and
daughters, yeah.
And I think that's the problem, is they're two different traits. When you're measuring methane on a growing bull versus the daughters, I think that was the criticism of that work initially, was that it's not the same trait. So I think we need to be really careful about which phenotypes we choose. So let's choose phenotypes on milking females, first of all, and let's make sure we have enough phenotypes to generate a reliable breeding value. And if you do that, you're going to get the right results. But if you don't have enough phenotypes or you have the wrong phenotypes, it's not going to work.
And I think it also, to me, highlights the value of validating what we're trying to accomplish, right? I think we haven't done enough of that historically in animal breeding, whether it be beef or dairy. We create phenotypes and we select for those phenotypes, but how often do we go into the commercial systems and actually validate that? Now, in dairy is probably a bit different because we collect a lot of data, but certainly in beef, we don't do a lot of validating of those traits that we've built. And so I think for me, it just highlights, Birgit, that exactly what Mike just said, right? Are we using the right trait? Are we using the right phenotype? Are we seeing, what are we expecting to happen when we actually implement that? And can we validate that that's actually what's happening? And I think what we're seeing in that is we can validate that that's not what we expected to happen is not happening. And likely it's because, we chose the wrong trait.
Just a comment. So I'm sure our New Zealand colleagues, they're looking carefully into those results that are available. And I know that there is a Norwegian study actually as well. So in Norway, they also measure methane on bulls and then look at the daughters, and there the correlation was actually high, was 0.6. So I think that's not the last results that we are going to see. So we have 1 minute left, and I would like to close the discussion with one question that we have is, the trait definitions. So I'll ask each of you, very, very brief, very short. What is your preference? What should we use? Start with Guillermo.
Yes, for me, as I show in the result, is residual methane intensity. So it's methane production in grams per day adjusted by energy in milk and body weight. So that is the trait that I will go for, because as I show, we don't have the strong correlation with fat, milk, and is positively correlated with feed efficiency.
I'd agree with that we need to choose a trait that, first of all, is not a ratio trait, because it's a lot easier to work with actual methane rather than methane intensity, at least at the genetic selection level. And I would do that after you correct for production and all the other expectations you would have for methane emissions. So we chose methane efficiency, corrected for milk, fat, and protein, but residual methane is also a good way to go. They're, I think, fairly closely related.
Obviously, our new traits have the embedded enteric methane prediction within them, and that's sourced directly from peer-reviewed research. So that's been shown to really help kind of influence some of the sustainability opportunity within our secondary milk methane intensity and some of our other enhancement traits. So I think as long as it has to be part of the conversation before you can even get to picking a specific trait to focus on. But really just thinking about the whole animal. Ultimately, that animal still is responsible for producing milk, and how we can help her achieve that goal is what's important.
So when it comes to commercial implementation, when I think about putting it in an index, coming up with the economic weight and those sorts of things, I'm not convinced we know what the right trait is, to be perfectly honest. I sort of tend to lean towards some sort of residual trait, but at the end of the day, I just don't know. I'm not convinced.
Great, thank you all. So thank you all again for the presentations and also the discussion. So I think it's highlighted nicely that genetics works. And as I said in my presentation, I hope everyone in the room does not leave the room without believing that genetics works. So thank you very much.